Hello. Majority of cancer patients suffer terrible side effects because majority of the cancer drugs are terrible. They not only can't kill the cancer cells, but they also target many normal cells. We clearly need better drugs. I'm interested in understanding the specific shapes and forms of molecules so we can get better cancer drugs. Now, microscopes are a very useful tool in science and medicine. Why? Because they help us to visualize cell behavior. And that's important. Sorry about that. And that's important because we need to understand how these diseases work. For that, we need to visualize these cells. Now, what if there was a way to color these individual cells so we can distinguish them better? And actually, there's a way to do that. To simplify a technique, you basically take your cells and color them up, say orange, and now you can see them better. But we need a fluorescent microscope for that. This microscope will fire laser beams at cells so you can look at the orange cell very clearly, like this. So in this experiment, I am tracking the movement and behavior of orange cancer cells that are surrounded by normal cells. So as you can see, this is pretty fascinating and quite clear. But cells are very complex. They contain thousands of molecules of very different shapes and forms, each performing a specific task in the cell. And some of these molecules may even team up together to perform the different tasks. How do we understand if two different molecules are interacting and doing a specific function? You can label them up. For example, let's say a molecule is shaped like a Pac-Man. You can color it green. Let's say a molecule is shaped like a triangle. Color it red. And now under the microscope, what appears to be red and green spots will give us precise information about the proximity of these two different molecules. In fact, we might even know if the green Pac-Man is going to chew up the red triangle. So I've shown you how to understand cell behavior using this coloring technique, how to understand the behavior of two different molecules with this coloring technique. What if you want to understand the behavior of one individual molecule? That's what I'm interested in. I'm interested in this molecule known as insulin receptor. This receptor is a bad guy in cancer. It misbehaves in cancer. What we know about this receptor is that it contains two identical parts that need to come together to perform its job. But what we don't know about this receptor is that after it comes together, what is the specific form and shape it can take in real time? And that's what I want to find out. How do I do that? Well, I color it up, just like that. But what I do is I'm coloring up the same individual insulin receptor with two different colors. Now I can watch the movement and form and shape the molecule takes by just looking at how these two colored parts are moving. But how does one do that? Using a super special single molecule microscope. This one will fire laser beams at the cells, in fact the molecules, and then you can visualize the movement of these colored portions. This microscope is designed to visualize only individual molecules. So although you may not be able to see the receptor per se, you will watch the individual colored portions pulsate as the laser beams strike it. So how does that look? What you can see in the screen is not a starry sky, but individual molecules lighting up as laser beams strike it. By alternating between red and green, we can actually get a lot of information. What kind of information? The kind of information is that these molecules are actually very dynamic. Those colored portions are moving in real time. And actually, we can get distances between these two colored portions to a very great accuracy. And that's important because you need to find a drug that will fit into the specific form and shape. That's important. And so by watching the dance of molecules, we can understand their shape and form and design better drugs. Thank you.